Fantastic. So our next speaker is Dustin Tran. Dustin is a PhD student in computer science at Columbia, where he's advised by David Bly and Andrew Dowland. He works in the field of Bayesian statistics, machine learning, and deep learning. In particular, he's made a lot of progress in approximate inference, and in the field of probabilistic programming, he leads the development of Edward, a library for probabilistic modeling, inference, and criticism. Dustin, thanks so much for being here with us today.
Should I speak in this mic? Okay.
Speak in this. This is just for
the recording and everything. Okay, perfect. Oh, I can do this. Hi. So thanks for coming, and thanks to the organizers for organizing this workshop, and of course, thanks to the reviewers for the high quality feedback. It was very useful. So I think one bit catchphrase for what I'm going to be talking about is that we wanted to use GANs for genomics. Now, to give a little more context to that, I think we as a community in machine learning have developed really powerful tooling for modern probabilistic models and subsequently highly accurate and highly scalable inference algorithms. And so Dave Bly, my advisor, and I, and many others in our research group were very interested in just trying to take these ideas and apply them to real-world problems. And so we sought this investigation in genomics and causality. We spent a year and a half. We struggled, and I think as a fruitful and interesting endeavor, I think it's just interesting to consolidate these sort of domain-specific knowledge and modern probabilistic models. And so without further ado, one way to describe this is to just start with a problem setting. So what are genome-wide association studies? You can think of them as consisting of just this very high-dimensional data sets where each data point is a single individual. And basically, with really powerful technology, you can basically sequence the entire genome of a person, and there might be a particular trait that you're interested in with respect to this data set. And the trait is formally a phenotype, and it may represent something like a metabolic level, the height, or a specific signal or indicator for a particular disease. The genetic factors are these things known as single nucleotide polymorphisms. They're particular loci within the entire genome, and you can think of them as being specifically encoded as 0, 1, or 2. And these are basically unphased data and represents a number of how many major or minor alleles you have at that particular location. And this is really amazing. The SNPs are displayed in this figure at the top. You can think of each of these columns, a slice along this vertical axis, as being one particular person and their entire set of genetic factors. And you might be interested in, like, what could you really do with this data set? Given our technology that we can sequence your entire genome, what is something you might try to answer? And genome-wide association studies are interested in trying to understand how genetic factors, these single nucleotide polymorphisms, cause traits, diseases in the individuals. And this is an interesting relationship. This goal allows us to understand something like, given a specific person's genetic predisposition, how likely are they to have heart disease at age 60? What is their height given just the set of genetic factors that they're encoded as? And not only can we do that, but we can do something like, if we have a particular genetic factor, if we manipulate this, we set this particular value to be another setting, 0, 1, or 2, how does that change the particular trait? And understanding how particular genetic factors cause traits is very interesting, because this is really one step in understanding how you can design drugs to counteract and counterbalance the diseases that are prevalent in the real world. Okay, there are many problems in GWAS, and I'm going to be focusing on just 2 problems. One of the problems is about how to build richer causal models. And so in this figure, if you just look at the middle column, there are a bunch of SNPs. There are M SNPs. They range on the order of hundreds of thousands to millions. And one causal relationship is understanding how X causes Y, the trait. And currently in the genomics community, we typically apply simple models such as linear regression. And these models apply a few to no nonlinearities. You might include some spline functions or polynomial lower order nonlinearities, or you might hand engineer interactions. You might say that this particular SNP and this particular SNP have this interesting interaction, and so I'm going to include it into my linear regression. What we'd like to do, because there are millions of SNPs, exponential in the blow-up of complexity and trying to understand all of those interactions. And in particular, in genome-wide association studies, there is this phenomenon known as Epi Stasis, which is the interaction between genes. And that interaction is very important, or recent studies have shown that that interaction is very important in understanding how those genetic factors cause those diseases. And another thing is this left column, which represents Z. And this Z here is a problem in just doing causal inference. And we have to adjust for what are known as latent confounders that influence or confound our inferences from X to Y. And there are two particular confounders that are particular in genome-wide association studies, and one is the idea of latent population structure, that there are subgroups in the population that all have ancestry differences. There may be a certain group in that have ancestry related to Asia, and there may be others that are related to Europe. And the data set is not just statistically independent, but you'd like to incorporate this sort of latent structure. And another is relatedness among individuals, where we might have sequenced not just taken people randomly from the world, but taken particular families. And so as backgrounds, we'd like to just describe first probabilistic causal models and then go on from there. And so one way to understand probabilistic causal models, which extend probabilistic graphical models, is in this top left figure. You can think that there is a global variable beta, and here I'm going to be writing all of the random variables as some noise source and then some deterministic function. And so beta here is going to be set as some function of a noise source that can be, say, standard normal or uniformly distributed. And then for each data point and in some data set size, you're going to say that there is a particular feature X, which is a function of its own noise, and some global confounder, some global variable. Finally, there is some trait Y, which is a function of its own noise source and its own feature vector and the global variable. And in probabilistic causal models, we're interested in estimating something like the causal mechanism f sub y. This determines precisely how all of the variables in this phenomena, captured as a probabilistic causal model in this graph, propagates and influences the traits. And so we might like to calculate something like p of y, given that we do or manipulate x and force it to a particular value, little sub x. And this is interesting. This is not the same as the conditional p of y given x equals x. And you can think of the conditional distribution as basically taking the joint. You have probability of y and x, and you're trying to filter to the subpopulation such that x is set to that value. And the reason why those two are different is because in general there may be phenomena, some latent variables that are forcing not just x to be that value, but are also causing y. And so we'd like to detach all of the things that can have confounded our inferences from y to x, so from x to y. Fortunately, under this causal graph, through theory that Judea Pearl and many others in causality literature have developed, we can say something, we can use this causal graph and leverage identifiability sort of structure. And so we can say that the distribution of y, given that we set x to that value, is in fact equal to the conditional. And this is really amazing because what this allows us to do is estimate the causal mechanism purely from observational data. We have collected all of that genomic data, and we can infer causal inferences just by learning the conditional distribution. And an example of this is an additive noise model. How does an additive noise model work? First, you have some trait, and you set it to be equal to some function of its feature vector and its global variables, and then you add some noise source. The function could be linear or use some spline functions, and the noise source can be something like a standard normal distribution. If it's standard normal, that induces a unit variance Gaussian distribution on y sub n. And then you can just put a prior on the parameters of the function class, and you can just proceed as usual. You do Bayesian inference, you can do maximum likelihood, and standard algorithms all exist to do this. Now, one thing we might like to do as an extension is to take that additive noise model and extend it with this nonlinearity. We like to just place a neural net on this thing. So you can think of something as implicit models as just positing distribution via its generative process. And just as the last talk described, you have some noise source and you define some non-parametric function g. And if you set this function g as a neural net, this really enables multi-layer nonlinear interactions. And in our work, what we showed was that you can leverage this idea, put non-parametric structural equations across all of the causal graph, and implicit causal models, which leverage these neural nets, are universal approximators of any causal model. And that's just a straightforward theorem from neural nets as universal function approximators of continuous Lipschitz bounded functions. Now, we'd like to use this idea for genome-wide association studies. So let's consider a generative process for how that arises. So first, we're going to posit some latent variable z that is shared across all of the SNPs, and that's what I'm writing here. And afterwards, we're going to write a generative process for each individual SNP, which is conditionally independent given that z. And so there's an individual function at g sub x sub m, and that is for each SNP. And then finally, we're going to say that there is a trait, and that trait is going to be a function of all of the genetic factors, the local confounder, and its own noise source. And then we're going to do this process for each person. For each person, they have their own latent confounder, they have their millions of SNPs, and they have a particular trait. So let's posit the distributions and the functional equations for all of these 3 lines. So for the confounders, we're just going to say that the z comes from a standard normal distribution. And there is some latent dimension k that we set as a hyperparameter in the algorithm. For the SNPs, this is a little more interesting. Essentially, we have this, in the data set, we have this huge matrix of n by m, the number of individuals by the millions of genetic factors, and we like to posit some generative process for that. The way we do this is to posit a binomial distribution from 0 to 0, 1, or 2, and we're going to say that the logit probabilities of that binomial are some nonlinear function of the confounder and these latent factors. And so in this figure, what I'm showing here is this old idea from genomics where they've applied PCA, principal components analysis, to basically project the millions of SNPs onto a lower dimensional space. And so what we're doing here is basically the exact same thing as PCA, but making it nonlinear. We're saying that instead of saying that there is an inner product of some low-dimensional latent factors in Z sub n, we're going to just apply a neural net to this thing. And then we're going to posit standard normal priors over W sub m, the latent factors, and our parameters to the neural net. And this generalizes logistic factor analysis, if you're familiar with that model. Finally, for the traits, what we're going to do is do the exact same thing with implicit models. We're going to plop down some neural net. It's going to be a function of all the genetic factors and its confounder and its own noise source. This generalizes linear regression. It's not additive with respect to the noise source, and it incorporates interesting interactions between the genetic factors and across with the confounders themselves. One interesting tidbit of this model is that we don't just use standard normal priors over all the weights and biases of the neural net. One thing is that with GWAS, the number of genetic factors is so large that it's just very difficult for a neural net in general to learn the signals. So what we do is we basically sparsify the first weights that go into the hidden layer. And we take this idea from statistics called a group lasso prior, which couples all of the weights that contribute to a particular hidden unit. And what this allows us to do is basically sparsify the weight matrix that goes into the first hidden layer, and this encourages learning from sparse inputs, because we expect a priori from our domain knowledge that very few genetic factors actually do influence a genetic phenotype or trait of interest. And so this is what allows us to learn that sort of structure. Now, for causal inference, if we'd like to estimate the mechanism f sub y, what causes that trait, we just calculate the posterior distribution over the function class, the theta. And how we do this is just doing the joint posterior, and we marginalize over this. And this is interesting. This accounts for that latent confounder, z, and what we're effectively doing is we infer the posterior over theta, the conditional posterior over theta, and we average over all of the posterior samples from z. So we're essentially inferring z, we infer theta given z, and we do this over multiple iterations. There is an interesting fact here with respect to causality, which is that, in general, when you do inference with latent confounders, it can be very dangerous, because you're using the data to infer the confounder, and then you use the data again to infer the causal mechanism. And in our work, we showed that this is in fact okay, because we showed that the posterior distribution over theta provides a consistent estimator of the causal mechanism, an asymptotic argument. Now, as an algorithm, the posterior distribution is intractable. And in fact, the model itself also admits an intractable likelihood. And so this bars traditional algorithms. And so what we used was an idea from modern probabilistic modeling. We used likelihood variational inference. This is a scalable variational inference algorithm that allows us to approximate the posterior distribution and not require the density of the likelihood distribution. And we use this idea to scale to millions of genetic factors. And if you're interested in the algorithm, it is in fact available as open source in the Edward probabilistic programming language. So as a simple simulation study, we took this huge collection of experiments from a few Nature papers, and we varied over a number of configurations. In general, trying to do causal inference and validating causal inference on observational data is just impossible. You can show that. And so what we did was we did this semi-synthetic sort of experiment where we took observational data and we sort of finagled them around and at least had true parameters that we can still leverage. And so we had 11 different configurations. They are on the order of hundreds of thousands of SNPs, and we ranged from 940 to 5,000 individuals. Each configuration, we had a replication of 100 different times. So in fact, there were 1,100 different experiments just in this table itself. And that's pretty massive. And what we showed was that across a bunch of different methods, principal components analysis, linear mixed models, and this GCAT, which does logistic factor analysis, we are significantly higher in accuracy. And what we're showing here is basically that our inferences are robust to spurious associations. We can infer what are the particular causal factors that are influence a genetic trait, and without saying that without saying that there are false positives, that there's a genetic factor that we predict that there's a genetic factor that causes the trait, but in fact it doesn't. And across all of our experiments, we've seen this with significantly higher accuracy. We've seen that leveraging modern probabilistic modeling really allows us to do so. And that's my final slide. Thanks so much.
Thank you so much, Dustin. Is there any question? We have 3 minutes for questions. Any questions at all? Sheil.
Have you considered so there is this hypothesis of linkage disequilibrium, which means that basically there is some collaboration between SNPs that are locally.
Yeah. So Jill just mentioned that there is this idea in genomics of linkage disequilibrium, which is that basically the genetic factors are not independent. There is correlation in between them, and we haven't corrected for that in our work. But I think that's also an interesting latent variable that's useful to try to account for. Kyle.
The group lasso that you talked about, I just want to, you still have to, you have all of the links there, but you're trying to force some of them essentially be 0 or not?
That's exactly right.
But in terms of the actual network or something, I mean, you still have all the communication and all.
Yep. In our neural net for the traits, it's a feed-forward network, and we only try to posit a prior such that we can sparsify the weights to the first hidden layer. But in general, it's fully connected a priori, and then post-inference, a lot of them end up zeroing out.
After you've learned the relationships, can you go backwards and actually see if those are sort of do sanity checks on to see if they actually make sense from a genetic perspective?
Yeah. So we apply this to a real-world GWAS, and in general, it's hard to validate. So what we did was there's a well-studied GWAS. We have a pretty good knowledge of what indeed causes diseases and metabolic levels, and we just checked whether or not we can infer whether or not we do recover those genetic factors. And in the full version of the paper, we showed that we can in fact do so. In the back. Yes. For the generative process for the genetic factors, it's just this huge matrix. And VAE is also known as nonlinear logistic factor analysis. And so we extended PCA to in the realm of something like a variational autoencoder.
question in the back, maybe?
Yeah, so on the simulation, like we were using like 2,000 people and 100,000 SNPs.
Yeah.
Modern GWAS will like densely imputed data will have 10 million, 15 million SNPs and 100,000 people. So I'm
Yeah.
wondering if you have thoughts on strategies for big neural networks when the data space is so high and you can't really fit all the memory, but the feature vector is really wide.
Yeah, I think this is really not the end of the story. I feel like we just got a preliminary step in understanding where modern probabilistic modeling could be useful. And I think it's something that we would love to do, which is scale this to a real-world genome-wide association study example and really show that we can infer genetic factors that people just haven't shown before, not just improve benchmark experiments. And I think there are a ton of open problems there, and I'm happy to chat about that offline. But many different issues many different issues appear in learning the neural net, and what are the right priors around the parameters and the local confounder, what is the right dimension, all of these things play a role. Thanks.
All right. Thank you so much again.